Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Derek Abbott. He's a professor at the University of Adelaide. He's the director of the Center for Biomedical Engineering, and we're going to talk about biologically inspired engineering. So, Derek, thanks for coming.
Thank you very much.
So if you would, tell me about your work.
Okay, so my background is I did both physics and electrical engineering. And electrical engineers are interesting beasts because they're trained to do a number of things, look at data, and mine that data, everything from there through to designing circuits, etc., etc. And in that vast span of capabilities of an electrical engineer, if you look around, say, hospitals and medical institutions, everything is electronic, all the diagnostic equipment, etc., etc. And so where does that all come from? That comes from electrical engineers. So electrical engineers, the particular branch that I've gotten into is very deep into what's called biomedical engineering. And so it's bridging the fields of electrical engineering with medicine and biology. And so as an engineer, one can actually try and tackle some tricky problems to do with biology and medicine. A good example is DNA, because DNA contains a lot of information, and the area of bioinformatics, which is to kind of mine through that information and extract what you need from the DNA. The skills of a computer scientist or electrical engineer are very important for creating, say, the software and the techniques that are involved in doing that. So, yes, it may seem counterintuitive, but engineering does have a lot to do with medicine these days.
Okay. Well, so what in particular that you're working on did you want to discuss and go into detail about?
So I thought we were going to discuss the Somerton man case, which is a particular project I work on, and is quite engaging and interesting.
Yeah, what's the background of it? Tell me. It's not like Piltdown Man and all, is it? It's a different man.
It's just, no. So you obviously haven't read anything about it. Okay. So one interesting example of a project I've been working on some years now, more than 10 years, is the case of the so-called Somerton man. What this is it's a man that was found deceased in 1948 in Australia. He was just lying on the beach. He looked like in his early 40s, was wearing a nice suit, and he was found one morning and he was just dead. The body was dry. There wasn't a scratch on him. There was no signs of any violence. And to this day, no one knows how he actually died and what his identity is. So it's been a long mystery, and it's engaged the world through means of modern communications and internet. This story has really gotten out there, and people are curious as to who this guy possibly could be and where he came from. And so I decided about 10 years or so ago to put my skills as an engineer, also with biomedical engineering, to have a look at this case and see if I could tease out some information. So to tell the story of this case, so the audience know why it's so fascinating, is this man was found on the beach. Police took him away. He was sent to a mortuary. He had an autopsy. The autopsy couldn't find any traces of poison at the time or anything like that, because that's what they suspected, as there wasn't any sign of any violence. But nothing was found. And they thought, okay, somebody will come forward and identify this guy, a missing family, a family member that's found him missing or something like that. And they waited and waited, and, month, 2 months goes by, and there's nothing. So they're beginning to realize that, hey, this is possibly a stranger from out of town, and no one's coming forward. So the mystery thickens. The police, using the hypothesis that he's from out of town, go and check the locker room in the local station where interstate trains come. And they found in the luggage room, they find an unclaimed case from that's date stamped, the date before he was found dead. So the timing was beautiful. They opened up the case, found some clothes and effects, and the clothes fit the man, and there was some thread in there, and it was exactly the same unusual colored thread that was used to sew some of his buttons on his jacket. So the police decided that this case was definitely his, but it didn't lead anywhere. And so they got another pathologist to look over the case to see if there was anything missing. And a guy called Bert Cleland, who also worked at my university back in the 40s, he was assigned to the case, and he noticed one small thing that was missed, and that inside the man's fob pocket was a tiny bit of rolled up paper, and on it had the words printed, not handwritten, printed, tamam shud. And no one knew what that meant at the time, but eventually someone came forward and said, oh yes, this is the last words in the book of poetry called the Rubaiyat of Omar Khayyam. And so it's weird. So they thought, okay, so this has been torn out of the book. So this is like a 1 in a million chance. They decided to advertise this in the local paper. And they said, has anybody got a copy of the Rubaiyat with the back page torn out? Please hand it into the police station. And I think they got something like 2 or 3 people hand in copies. And it turned out that one of them was the right one. There was a match. And what was interesting about this copy that was handed in, they asked the guy, where did this come from? And he said, this is 6 months now after the death. He says, about 6 months ago, I just found it, in my car. And don't forget, in those days, in the 40s, people, left their cars unlocked and windows down and things like that. And so it's almost like a passerby had just tossed it into the car.
But it was in the guy's pocket? Like was he wearing a suit or?
So we're talking about the guy who found the book in his car.
Oh, but I thought you said the body himself, the guy that was dead, he had this message on his person.
Oh, he had, yes. So this tiny piece of rolled up paper with the words tamam shud, Bert Cleland, the pathologist, found it in rolled up and tightly jammed down his fob pocket of his trousers. So for those who don't know what a fob pocket is it's a tiny little slit that's in the band of your trousers. And in the old days, this was used to put your fob watch. But we still have these fob pockets today, and in modern trousers, if you look carefully in men's trousers, and most guys don't even know it's there and don't use it. And some people use it to put a coin in or something like that. So, but anyhow, this piece of paper was found. They realized it was from the Rubaiyat of Omar Khayyam, so they advertised that in the paper, and then 2 or 3 people came forward with books they had found with the back page torn out, and one of them turned out to be the right one. The police had carefully examined the torn out piece of paper with the suspected correct book, and they found that the texture and weight of the paper exactly matched. and the words were torn out of the right place in the book. So they were convinced they had found the right book, and the guy that handed it in said that it was tossed into his cars 6 months earlier. So, and 6 months earlier at the time was when the man was found dead. And in those days, wouldn't be that unusual, because in the 40s in Australia, people just left their cars unlocked and the windows down and things like that. So it's a bit strange. That was a 1 in a million chance that this book came forward. Now, the interesting thing in the book is it had some strange incoherent letters penciled in on the back, and some phone numbers. And the police at the time thought that maybe this is some World War II, Cold War, some World War II kind of spy code or something like that, because they were just like jumbled up letters. That's how it looked. But no one could crack the meaning of these letters. And there was a phone number there as well. And that phone number led to a local lady who lived literally 5 minutes walk from where he was found dead on the beach. They interviewed her, and she claimed no knowledge of the man. She said she didn't know who it was. And but the police noticed she was a little bit evasive. They didn't. The case was never treated as a homicide. They always thought it was some kind of suicide. And so she was not pushed any further because it wasn't a homicide they were looking at. So she was left alone, even though they noticed her behavior was a little strange and evasive. And they kind of suspected she knew who this guy was, but she wasn't saying. So as an engineer, when I read about this case back in the 90s for the first time, I thought to myself, this is really interesting. As an engineer, I would like to look at these strange letters on the back of the book and see if we can work out something about them. I didn't feel that I would necessarily be able to crack what they mean, but I was thinking I could at least eliminate what they're not. So because engineers are into things like cryptography, when you do banking transactions and things like that, you have to encrypt the information, and also in computers when you transmit information. So we as engineers, we know about that kind of stuff. So I thought, okay, what we will do, I'll set this as a project for some of my students. And it was around 2007 I started doing this, started looking at this code and setting it as a project for my students. And we eliminated a whole bunch of things. So we looked at all the kind of ciphers that were used around World War II and before. And we were able to write computer programs and do statistical analysis to show that these sequence of letters weren't any of these kinds of codes. And our statistical analysis showed that it was most likely simply the first letters of words in the English language. So it's almost like an aide-de-memoire, if you like, just using first letters of words. So what those could mean could be absolutely anything. So I kind of dropped looking at that for any further, and I decided that what was really interesting about this case, because it had engaged me by then, was finding the man's identity and trying to dig into that further, because to me it seemed that finding how he died was a little bit out of the question now, so many years later.
Well, did they do it? They didn't do an autopsy on him back in the day, right? They just buried him.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Yeah, as I said at the beginning, he had an autopsy, but they were unable to determine if there was a poison, and they were unable to determine if any cause of death. So
they had no suggestions at all. They couldn't. Did they do any photographs?
No autopsy photographs. The only photographs they took were of his face, so that they could be sent to the press. so that and published in newspapers, so that in case anybody could identify him. So back in those days, they weren't really big on doing autopsy photos as such.
Yeah. Is the body exhumable, or is the body gone?
The body is buried in a cemetery here in Adelaide, and it's still here today. And so it is potentially exhumable, though the state that I'm living in is fairly conservative when it comes to giving approvals for things like exhumations. So it's very difficult getting an exhumation here, say, compared to America. We're way more conservative. So there's an issue there. So what I did is I looked into the guy's effects, and it seemed to me that the police had reported that the man had a jacket that he was wearing that had stitching that was only producible by machines that were in America at the time, not in Australia. We didn't have those at the time. So that was an interesting bit of information. And the police said the man had an aluminum comb that was only associated with US troops at the time that were in Australia. So that was interesting. But those two items aren't really enough just to say, hey, this guy's American, but it's an interesting clue. And so I looked at his effects further, and I noticed something strange about his tie that he was wearing, which is in his photo, which is still shown in his photo, and that his stripes of his tie go the opposite direction to the way striped ties slant in Australia. And I looked this up because I had no idea what this meant, and I looked up the history of ties, and this was quite interesting. I found that the UK and Australia traditionally had striped ties striping in the direction what is called heart to sword. So your sword is on the right side of your body, usually, because most people are right-handed, and your heart is on the left. So the stripes go in that direction. And then I read that stripes go in the opposite direction in America. And this tradition started around the 1920s in America, because in the 20s, America started to import tie fabric from the UK and decided to cut the ties in the opposite direction to be more distinctive from what was the traditions of England, to separate from them. And so when you look at old photos of US presidents, they always have striped ties going in the opposite direction to what we do. Except for your latest President Donald, he has stripes going the wrong way, which is very unusual for a president. I noticed every single TV item that he's been on, he's had stripes going the wrong way, except for this latest presidential debate with Biden. He suddenly has stripes going the correct American way now, which is interesting. Anyway, that's a sidetrack. So, so this kind of making me think, could this guy be American? So it was interesting. So then the next thing I did was I looked carefully at the inquest, which is reported, and we still have that today. I should mention that the poetry book and the autopsy report, all these things have been lost now through the passage of time. The only thing that actually exists still today is at least the court inquest transcript. And in that transcript, it basically, when the pathologist is giving evidence, he basically says that the man's lateral incisors, that's these teeth that are next between your canine teeth and your central teeth, he said they're missing. But he then adds, if the man were to smile, you wouldn't notice any gaps. So what that means is that it's symmetrical. There is both lateral incisors and there's no gaps. So this is suggestive of the fact that perhaps his teeth had grown like that. So it's not like he lost them because there were no gaps. So I spoke to dental experts at my university, and they said, yes, this is a rare condition. People do grow up with missing teeth. And to have two symmetrically opposite ones like that, the lateral incisors both missing with no gaps, is quite unusual, is quite rare.
Yeah, what causes it? Is there any particular reason?
It's a genetic thing. So this is then what struck my curiosity. I thought, if it's hereditary, this is a very useful bit of information. So I put that piece of information aside, and then I noticed something else about the man's photo. I noticed there was something strange about his ears, but I couldn't put my finger on it. I didn't know how to articulate what was wrong with his ears. So I went to an anatomist at my university who happens, just by luck, to be an expert on ears, of all things. And I said to him, these ears, I said, this doesn't look right. Can you, what is the correct words in anatomy to talk about these ears? And he said, yes, you're right. The ears are very rare, and it is an hereditary trait. And he said the way to talk about it is, he says that the upper hollow of the ear is enlarged and the lower hollow is small. Whereas in normal people, it's the other way around. If you look at your ear, you'll find it's the upper hollow of the ear that's the smallest one, and the lower hollow is larger. So his ear is kind of inverted in the way the hollows work in the ear. And he told me that this is hereditary, and I was able to dig up scientific papers on this and confirm that. So
people that have that, does that coincide with the strange arrangement of teeth as well, or the missing incisors?
No, there's no correlation that we know of.
Too bad.
Yeah. So, and there's no kind of ethnic correlation with these things that we could see, particularly by looking at the papers that are written on these things. And so what we did with these pieces of information, I thought to myself, this is very interesting. So if we're looking for a relative, perhaps a relative might have these two genetic markers, so to speak. So the next thing I did was look into the identity of the woman who was evasive, who the man had her phone number of, and she was found dead. Living 5 minutes away from where he was found dead, 5 minutes walk. And so I found her identity, a little bit of detective work, because it wasn't generally published in the newspapers at the time. It is now, the name is now public. Her name is Jo Thompson. And so I found out about her, looked up into her background. And unfortunately, she had passed away in 2007. And I had found her identity in 2009. So I missed her by 2 years. So that was a shame, because I was hoping I could talk to her. So I then thought, well, does she have any descendants? Maybe I could talk to them, because maybe she told them something. So that was my line of thinking. So I found that she had a son, and his name was Robin Thompson. And unfortunately, literally by the time I had found his identity, he had died literally 2 or 3 months before I found out who he was. So I just missed him. Oh. So I was really sad about that. Obviously, he died because he died 2 years after his own mother. He obviously died young. He had cancer. So, but when I found his identity, I found out a little bit about him, and apparently he was a professional ballet dancer. And that was very interesting, because when I went back to the inquest documents, it does say that one of the features of the man that was found dead is he had extremely well-developed calf muscles that were high up the calf. And so there was speculation at the time in the 40s that one of the possible occupations of the man is maybe he was some sort of former dancer.
Oh, okay.
And so when I found out the son's a dancer, I thought, oh, this is interesting. And then I found that Jo Thompson wasn't in fact married at the time that she had the child.
How old would the child have been when the guy was found dead?
He would have been about 1 year old. So the timing perfect. So I'm thinking to myself, okay, so what if this is the son of the Somerton man? Now, the nice thing about him being a professional ballet dancer is there were lots of photos of him in newspapers of the time. So he was quite a good ballet dancer. He had danced for the Australian Ballet Company. He had danced for the ballet company in Basel in Switzerland and the Royal New Zealand Ballet Company. He had toured America in the 70s with Rudolf Nureyev. And so there were photos out there that I was able to obtain. And so when I looked at these photos, I noticed, lo and behold, he's got these lateral incisors missing with no gaps. So similar ear type to the Somerton man, with the upper hollow very large and the lower hollow small. So I thought, wow, that's an amazing coincidence that he's got both those features. His mother is single, and she's got this, and this guy's got her phone number. So I'm kind of joining some dots here. So there's either a connection there, or it's absolutely an incredible coincidence. So what I thought is, what we got to do is we've got to get at the Somerton man's DNA, because unfortunately we don't have Robin's DNA because he was cremated when he died. However, I have been able to reconstruct it, and the way I did that was I found that he had a descendant. So he had a daughter that I managed to track down. And this is a kind of a twist to the story. About 10 years ago, we ended up getting married, but that's another.
Oh, really?
Yes.
Wow.
That's a funny twist. But anyway, so what I was able to do was get my wife's DNA and her mother's, and basically subtract the two to get her father's DNA. Subtracting isn't the correct word. The correct terminology, as we say, is we are phasing the DNA. So we're phasing out the mother to just get the father's side of the DNA. So that was a way we've been able to reconstruct that. So then my next step was to try to get the Somerton man's DNA. So this is what we've been trying to do over the last few years. And it so happens, there's a part of the story I haven't told you, is that before the Somerton man was buried, the authorities of the time decided that they would like to make a plaster bust of the man in case somebody would come forward wanting to identify the body, and they couldn't because the body was buried. They could at least show a plaster bust.
That's unusual.
Yes. So they got a guy from our local state museum who was expert in doing strange things like this. His name was Paul Lawson, and I think Paul Lawson is 101 today, and he's still alive. He's still got all his marbles. I've interviewed him many times. A great guy. And he molded the plaster bust directly off the dead body. So he did it in two halves.
Were you able to ask him if he had any memory of things he noticed about the body?
Yes, I did. So Paul Lawson said that the man had exceedingly big hands. So he had average to small size feet for a guy that's kind of like 5'11" in height, but his hands were absolutely enormous. And I said, well, how enormous do you mean? And he said, Paul Lawson showed me his hand. And Paul Lawson himself has huge hands because when he was younger, he used to be an amateur wrestler. And so he was a pretty big guy himself. And he said, well, if you were to put the Somerton man's hands up against mine, I would say that, they'd be an inch bigger. So those are big hands. So yeah, he noticed things like that, which were interesting, but that hasn't led us into any particular direction. So the nice thing about this plaster bust, though, is that although it didn't lead to any identification, it still exists today in a museum here in Adelaide. And so I've been able to examine this plaster bust, and I noticed that it's still got hairs stuck in it in the plaster. So this is really interesting. Because it was molded directly off the man's body, the hairs are still embedded in there. And so it's a source of DNA. I found out that a policeman had a few years earlier tried to pull hairs out of this bus and have them DNA tested and didn't get anywhere with it. But what he had done is he had just basically pulled the hairs and, at that age, they just snap and, the roots are embedded in the plaster. And because this is now a historical object that's in a museum, you can't destroy the plaster bust. You'd like to basically it's made of plaster of Paris, and you could, in theory, just put a little lemon juice and soften it and pull the hairs out, but you couldn't do that now because we're dealing with a historical object. So what I did is I got a specialist at the university who specializes in doing DNA testing of ancient hair samples, like woolly mammoths that are found in ice and things like that. And she has had a lot of expertise with hair and was able to do things I wouldn't think of doing. And so I brought her in to look at the plaster bust, and she used a very clever technique to actually pull out some hairs by their roots. Basically, what she did is look for hairs that were clustered together. And if a hair was in the middle of a cluster, she could slide it out and not be restricted by the plaster. So she went very carefully with a magnifying glass around the plaster bust and was able to extract some hairs like that. So we got a number of hairs with roots. We also got some which snapped off, unfortunately, and so we got some plain shaft as well. Now, what we are able to do then, because our DNA lab at our university is geared up for doing ancient DNA of old archaeological samples and things like that, there's the expertise there to extract DNA from old samples like this. So what we did is, I think it was about slightly over 5 years ago now, we got one of the hairs with a root and did an extraction, and our lab got very little out of it, but they were able to get one thing. They found the maternal haplogroup of the Somerton man's DNA was in the H group, and that's all we're able to get. Weren't able to get any DNA sequence at all. It was all like a mess. So, and the reason for that is the concentration levels that they were dealing with, that they were able to extract from this hair, were extremely low and were below all the thresholds needed to DNA sequence from this hair. So we then made another breakthrough with the hair 5 years later, which was about last year now. What we did is we decided to take 3 of the hairs that had roots now, and instead of just 1, to try and up the concentration level. So we had the benefit of now 3 hairs and the benefit of the techniques being more sensitive than 5 years earlier. And this was really interesting because we've now got the whole mitochondrial genome, got the whole mitochondria, which is fantastic. And but there's a good and a bad to that. So the good news is that what this means is that the DNA is extractable from the hair. It's very difficult to do, but it's been done. And it shows that even though the man was embalmed in formaldehyde before he was buried, we actually got this clean mitochondrial DNA trace sequence that was unaffected by the formaldehyde. So that, because you see, there were doubts whether we could actually even get any DNA because the formaldehyde would have corrupted the DNA. But what this is showing is that the formaldehyde embalming process was patchy in this case, and there are patches of his body that were missed, which is good for us, good news for us. Even if we were stuck with a completely formaldehyde corrupted set of DNA, there are now modern techniques able to solve that problem, but it makes the problem even harder. Now, the downside of the mitochondrial DNA is it's exactly the DNA that you don't want. It's the DNA that actually doesn't tell us anything much. Because if you think about it, he's a man and his mitochondrial DNA isn't passed down to his descendants. So it doesn't help us with getting a match between Robin and the Somerton man. And it doesn't help us going the other way. It doesn't help us to identify who the Somerton man is. So one thing I should explain is that if we had what are called the autosomes of the man's DNA, and these are basically what the autosomes are basically they contain a mixture of DNA from both parents. Okay. And it's this part of the DNA that is actually used by DNA websites like 23andMe and Ancestry. And so these are websites that people like you and me can just for, say, $100 decide we're going to swab ourselves, put our DNA on these websites, and it's like the Facebook of DNA. And so what we can do is...
One quick question before that. With the mitochondrial DNA, wouldn't that potentially allow you to match someone that was his mother or his maternal line?
All right. So, yeah. So can I just finish my train of thought and I'll get back to that point?
Sure.
Can I do that? So what I was basically saying is that, so the way we would propose to identify the Somerton man is to actually make use of these websites. If we had the right part of his DNA that's inherited from both mother and father and not just mother, we could go put his DNA up on these kind of sites, and we could find distant cousins of his. And through the family trees of those cousins, we can triangulate back to him and find a family tree where there's somebody that's gone missing in the 40s, and voila, that will home in on him. So that's the idea. And this technique is known, it's called forensic genealogy. And this is the exact technique that adopted children use to find who their real parents are. So just by using your own DNA, you can trace your own parents using this method. And it is a method that is being used in the USA today to solve cold cases as well. And there has been some success with that in the USA. There have been some big cases cracked, like the Golden State Killer case recently was one that used that technique. So I thought of doing this, 10 years ago, before this, these techniques were really known and had become hot. But we didn't really have the technology 10 years ago to extract DNA from hair, from old hair like that, to get the whole genome. And even today, we are only on the edge of it, as I'll explain in a minute. So that's the downside of us only getting the mitochondrial DNA, that we're not able to put this DNA on one of these genealogical websites and find his cousins. We really need the autosomes for that. Now, going back to your question, why can't the mitochondrial DNA help us at all is because what that does is it finds anyone that's not only anyone that's connected to his mother, but it finds anyone connected to his mother's mother and mother's mother's mother and so on. Because it's what that DNA represents is the whole maternal line going back, who knows, 2,000 or 3,000 years. And so anyone that's descended from any of those women over the last 3,000 years will be a hit. And so you'll get thousands of them. So that's the problem, why you can't use that.
But if you correlate it to a period of time, and you can say, okay, this is at most 3 generations back or 10 generations back, and that would give you like a smaller subset or a pool of people that it could be. And maybe you could find out who their family is and trace it forward like that.
Yeah, it's very difficult.
Right.
You're basically dealing with such a large data set here that's a very tricky proposition, what you're suggesting there. What you really need are the parts of the DNA that are used on those genealogical websites, because the nice thing about those is they basically connect you quite nicely to anyone that's a first cousin, second cousin, up to about fourth or fifth cousin. Once you start getting to fourth or fifth cousin, though, even that is very distant and very difficult to do forensic genealogy on. You really want to find third and second cousins. Once you get down to that level, it becomes a lot easier to identify who someone is. Fifth cousin is almost like a stranger. It's a very tough problem. So that's where we're at the moment. Now, I just want to say that the DNA that goes up on these genealogical websites like 23andMe and Ancestry, they use something like 800,000 markers, and these markers are called SNPs or SNPs. And these SNPs are small segments of DNA that actually give information about the person's physiology, about, say, the color of their hair and eyes, their ethnicity, perhaps genetic diseases they've got, and things like that. So they give a whole ton of information about the person, as well as the potential to connect to distant cousins and even close relatives if you're lucky enough to find any. So that's enormous. And if we now compare that to, say, what police use for catching a criminal, the standard DNA kit for forensic tests in criminal cases is they usually only use somewhere around 20, it depends on which country we're talking about, but usually somewhere around 23 markers. And they aren't even these SNPs. They're a different concept. They use what's called STRs. So these STRs are kind of repeats in the DNA. It's a sort of, it's almost like thinking that it's like the DNA has stuttered, if you like. So they look for these stutters, 23 of these stuttering markers in the DNA. And this is how you, match a criminal to some DNA left at a crime scene. And this is a completely different ball game from that. And with the crime scene DNA, it's very unforgiving because you're only looking at 23 markers. So there isn't any room for error. You don't want your DNA to be contaminated or there to be any losses, etc. So you've got to be very precise, and they're very precise protocols with those sorts of forensic DNA analyses. But in our case, when we're doing the genealogical type approach, what people don't understand is because we're now dealing with 800,000 bits of information, it's actually more forgiving. There's more room for error. In fact, there have been cases solved in the USA where the DNA has been degraded so much that the investigators didn't actually have all 800,000 of those markers. They only had 10% of them. So they only had about 80,000 of what they needed, and yet they were able to do an identification and get what they need.
Gotcha.
So it's a much more forgiving technique, and think of it like this: if I say transmit a computer code, a piece of computer code across the internet, and there could be just one tiny transmission error, and you are the receiver, you try and run that piece of code, you run this program, the whole program won't work just because it has one tiny error. It's dead. Whereas if I send you a video across the internet and you watch this video, and this video has, hundreds of errors, bit errors, it doesn't really matter because, this video has got millions and millions of bits of information and, a few hundred, random errors here and there. You don't even notice it. And in engineering, we have a word for this. So we call it graceful degradation of information. And so with this whole genealogical approach, we're dealing with a graceful degradation. And and it's this concept that unfortunately authorities and police jurisdictions around the world don't quite understand that yet, that this is the case and this is a different ball game. And so this is why we're not seeing a lot of police jurisdictions around the world rapidly taking up this new kind of technology. But as it gets more and more out there and people understand this concept of graceful degradation, I think we'll see more of this in the future. So on this point, what we were able to do is from this hair, we did in fact last year actually get some SNPs, SNPs from the Somerton man's hair, but we only got about 2% of the 800,000 that we need. And 10% is the lowest amount anybody in the USA has ever used to actually crack a case. And so we're about 5 times lower than that. So we're really kind of struggling here. So there are possibilities that, we could run the tests again, in a few years' time and get a much bigger percentage, or we could get exhumation before then. But
well, I get a quick.
I feel it's
here.
Within reach. It's within reach, and we can get there. Sorry, over to you.
Yeah, that's right. A lot of threads here. Have you spoken to your wife about her parents, and if they ever said anything? Did she ever ask her dad, which was this person's son, about his parents and all that? I mean, what did you get from that?
Yeah, so basically it seems that her dad didn't know anything about this and assumed that the father that raised him in the end was his real father. And as far as we know, that's all he knew to the day he died. Okay. Also, we know that he didn't even know about the Somerton man case. He only even heard about it 1 year before he died.
Oh, wow.
So from information I've received from the family. So yeah, he wasn't in the know. And he was the sort of guy, probably in real life, that if he had known, he was the kind of guy that would bury his head in the sand and probably not want to know about it.
Gotcha. Yeah. These phenotypical abnormalities, the ear having, I guess, the reversed or a different kind of hollows than normal, overly large hands, strange dental structure. Do they correlate with any diseases? Is that known today, or do they correlate with any other, high blood pressure, low blood pressure, any other things that may give you a clue as to how he died?
No, we don't know if there are any correlations between these features and different diseases. There are many correlations between SNPs in people's DNA and different genetic diseases, and these are very well known and documented. So once we do get his whole genome one day, we will be able to look at all his SNPs and see which genetic disorders he had a predisposition to. So that will be a possibility. Though, many, perfectly fit athletes even today just wake up one morning and are found dead. It still happens today. And the usual reason is that they've had a heart murmur and they just pass away. So this can even happen to extremely fit people, and it's possible something like that happened to him too, because he was noted as looking extremely physically fit. He had a very narrow waist and strong, broad shoulders. And for his age, that looked pretty good. Now, but trying to correlate other features like sizes of hands and things like that with different disorders, that's an interesting question. Work has not really been done on that in the research world to kind of correlate all these features.
Well, the reason I ask is he had 3 at least very distinct features. So within one of those,
now in terms of research, these things haven't been researched, so we don't really know the answers to those. But what I want to bring this point up, because what you've raised is a very important question, and this is something that is happening all in the area of biomedical informatics. Because what we're seeing now is in the research world, a huge surge in the use of AI and deep learning techniques on big medical data sets. And so I think we're going to see more and more of this sort of happening over the years, and we will find correlations between different features and different medical disorders by using what's called big data and using deep learning on big data. So I think that's yet to come, and we will see more of this in the future.
It's a strange question. What's more important to you if you could only have one thing, to know how he died or to know who he is?
That's a good question. And I've always said to myself, what's more important is knowing who he was. I'm not really interested in how he died.
Oh, really? Okay.
Because in a sense, it doesn't really matter. What really matters is somebody's name, because once you've got somebody's name, you can pull out their whole history. You can find out who they were, what they were doing there, etc., etc. If I just found out how he died and didn't have his name, it wouldn't be that interesting. But that's my opinion.
Yeah. So now going back to the beginning, I see it with different eyes. What year was he found?
1948.
So how would he, and he was found in England, if he was American, were there travel logs of, if someone came from America to England, it would have been a very difficult trip. There wasn't really any commercial aviation or anything. So have you looked there?
Yes, there are plenty of ships' passenger logs, but it's like a needle in a haystack. That's a very difficult problem to work through those.
Oh, okay, even with all these distinct features, and there probably wouldn't be a lot of, I don't know how many people were coming over, but
yeah.
Interesting.
Don't forget there were a lot of American troops around in Australia in the 40s. So there would have been quite a big, there'd be quite a lot.
Okay. So one of my last questions, what's required to get an exhumation? Do you have like a checklist and you're hunting down or you're getting all the requirements so that he could be exhumed, or at least a tooth taken from him or something, or a hair from his head?
So are you saying if we were to exhume him, what would we use? Would we use the tooth or something? Is that what you're asking?
Well, two questions. Is it even possible to have him exhumed? If you do A, B, C, D, E, or prove this or that, can you then have him exhumed? Like, have you examined the legal method by which you could get it done? And then if you did, what would you take? What would you do? What would you look for?
Okay. Yeah. I think everything is in place and an exhumation could be done. It's just that more than one different authority has to be satisfied, and then they're kind of getting all those clay pigeons lined up, so to speak, is a difficult act. It's just difficult over here. It's just the politics of how it works here. So now, given that, let's say hypothetically we did do an exhumation, what are the sorts of things that could be done? So probably one could, do a whole body scan, a whole CT scan of the body, because it would be good to get his whole dental configuration recorded, and any fractures in his bones that might give a clue, because if one looks at old war records, etc., if it correlates with a known injury that somebody's previously had, that this can help with identification. We can do what's called bone isotope test, and this can locate where somebody was born. So that's also useful information. So that looks at strontium isotopes. I'll come back to the point about strontium in a minute. There's something else I forgot to tell you. But, and then, of course, there's the DNA. Now, we probably wouldn't get DNA from this particular man's teeth, because it's recorded in the inquest that his back teeth are missing. He's only got his front teeth. And usually when you want to extract DNA from teeth, you usually go for the molars, not the incisors. So what might be better is to use a piece of inner ear bone or something like that to extract his DNA. And then, assuming one can get those 800,000 SNPs that you need to go on a genealogical website, that would be a sure way, the great pathway to finding his identity. So those are the kinds of things one could do with an exhumation.
Since your wife essentially is his granddaughter, probably, is there any way to assert that and to speed along the exhumation or make it more possible because of that?
Possibly, one could, like when we do our next test from hair, and hopefully we'll get more than 2% of the SNPs we need next time, even if it's not enough, it might be enough to show that there's a correlation with my wife. But we'll wait and see. These are open questions. Now, so I want to bring out a couple of scientific points here. Listeners might be wondering why it is that we can get this whole mitochondrial genome so fantastically, and why it is that the nuclear DNA, the other parts of the DNA, are so difficult to get at. Well, what's actually going on there? And what's going on there is it's just the luck of nature that human cells tend to have many copies of the mitochondria. So each cell has, perhaps over 1,000 copies, whereas the nuclear DNA there might be only 2 or 3 copies. And so when we're dealing with old hair samples, and we're only using 3 roots, the concentration levels we're getting are very small for the nuclear DNA, but for the mitochondria, because there's more copies of them, we're getting just enough to get the whole mitochondrial genome out. So I just wanted to explain that's why there is that difference.
Are you able to revisit the plaster cast and extract more hairs? If you do so, do you think you'll get more of the SNP profile?
Well, I took the precaution of, when I first went to go to the bus, to actually get 50 hairs. So I've still got, over 40 left, hidden away. So hopefully I won't need to go back. If I do need to go back, hopefully I will be able to access it again.
Okay.
But because of the way, things change, rules change, with time, who knows whether one will get access again. So that's why I took the precaution of taking a lot of hairs the first time.
Well, if you opened it up and then restored it to look, to the eye to be the same as before, I mean, why would that not be allowed? It's historically significant, but then again, for people to appreciate the significance, is a lot is in the looking. If you were to open up a small spot that wasn't in normal view and then restore it, would that be a way to get into the interior of it somehow?
There isn't any difficulty with getting the hairs out. We do it by pulling in a very careful way, as I explained previously. I was just talking about whether one can even access the bust in the future. Who knows what will happen to the rules? Rules change.
Right, yeah, they do, yeah.
So that's why I took the precaution of making sure I got 50 in the first place. So we've still got 40 hairs to work with. Now, not all of those 40 have got roots, but I'm hoping we've got enough to do something with it again in the future.
What does your wife think about all this? That it's likely to be her grandfather and everything. Does it?
She's agnostic about that, whether that's her grandfather or not. And her attitude is, well, she would like this all to go ahead because she would like to know what the truth is either way.
What about yourself? Do you feel like you just need to see this through to the end, no matter how long it takes, or are there? Why have you spent so much time and effort on this mystery? What's captivated you by it for so long?
Well, it's like any research project that we do in academia. What makes a good researcher is you grab your topic and you see it through to the end. You persist. If we didn't persist, we wouldn't get anywhere. And the way I see it is that the techniques we build up along the way when we're solving a very difficult case like this to identify somebody, the software tools we develop, the techniques we develop, these are all goodies that can be used for future cases. There's lots of cases in the world where we need better tools for human identification, because don't forget there are natural disasters, tsunamis, all sorts of things that are happening in the world all the time where human identification is needed. And this is not just one isolated case. It's an extremely difficult case, and that's why I like it, because it's through a difficult case that the science gets honed and improved, and we develop the engineering tools we need.
Oh, one last thing that came to mind. Have you looked to see if there's missing persons cases, from the U.S. about that time?
No, we haven't done that. That's an interesting thought, but I don't have the resources myself to look at that, because that'll be a huge number when you look into that. If I was to get some assistance to look at that sort of thing from somebody in the US, I would certainly welcome it.
Okay. Well, very good. Derek, what's the best way for people to find out more about, your work in general, and then to find out updates on this mystery as you work on it?
I guess my website.
Okay. All right. So we'll include that in the show notes. And any last comments? I mean, it's really cool to talk to someone that's working on a mystery. You even have the voice for it to narrate it, so it's been a cool experience to interview you. But any last comments? Any things you want to mention before we go?
Oh gosh, you put me on the spot. I can't think of anything. A last comment. Yeah, I'd just like to say that I think this work kind of illustrates what we do in the academic world in a university. What we do is we work on problems, difficult problems, and hopefully in a multidisciplinary manner. And the problem itself isn't necessarily the goal. It's the kind of tools and techniques that we pick up along the way to help society as a whole. And I think that's a message about universities and academic research and how we operate. I think it's good for people to know that.
Yeah, no, it's true. Cases like this make you really work your hardest to figure them out, and in doing so, like you said, these methods could be used on other cases to help other people. And in all kinds of ways that we can't even imagine. So it's really cool that you're doing this work.
Thank you.
Well, very good, Derek. Thanks for coming on the podcast and talking about this, and I appreciate it.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.